Matt, let me know.
Yeah, just should be ready in one. Okay, we're good to go now. Go ahead.
Ready? Okay. Welcome to AFO Cafés. These are informal science conversations about all things birds. I am Valentina Ferretti, and I'm the current president of Association of Field Ornithologists. And I just want to tell those of you who are new to AFO Cafés that AFO Cafés are free and are open to the public. For those of you who are not members of AFO, I'm going to give you a brief introduction to our organization. AFO was founded in Massachusetts in 1922 as the New England Bird Banding Association. And the Association of Field Ornithologists is one of the world's major societies of professional and amateur ornithologists that's dedicated to the scientific study and dissemination of information about birds in their natural habitats. AFO encourages the participation of amateurs in research and emphasizes the conservation biology of birds. We also have a strong focus on Latin America, and we do this through meetings, grant programs, and outreach. Members of AFO receive access to member-exclusive events and funding opportunities for research. They also receive discounted registration for our vibrant annual meetings and assistance with animal care and field research permits through our continued participation in the Ornithological Council. So AFO was founded in 1922. That means that this year we are celebrating our 100 years. And we are going to be meeting, we're going to have our annual meeting in October in Plymouth, Massachusetts. And we are organizing a very exciting program. So we have invited 4 plenary speakers. Dr. John Kreischer will give the keynote address. He has taught ecology and ornithology at Wheaton College in Massachusetts for 48 years. He's also a past president of the Association of Field Ornithologists, the Wilson Ornithological Society, and the Nuttall Ornithological Club. Many of us also know him from his books. He has authored 10 books, including the new Neotropical Companion, and his most recent book is the Peterson Reference Guide to Bird Behavior. Scott Widensall is a researcher and author, and he spearheads several major research projects focusing on bird migration. He has written more than 30 books on natural history, including Living on the Wind Across the Hemisphere with Migratory Birds. This is a Pulitzer Prize finalist. We also have invited Dr. Emily Choi. She's a postdoctoral scholar in the Department of Natural Resource Sciences at McGill University and Environment and Climate Change Canada. Dr. Choi is studying the physiological response of thick-billed murres to climate change on Coates Island, Nunavut. And she's also one of our council members. And last, Catherine Parsons will talk on Thursday, October 13th, and she has 45 years of experience in coastal waterbird research management and policy. And her career began as an intern at Manomet Bird Observatory, where she later served as a senior program leader and director of the field biology training program. But in 2011, Dr. Parsons became the director of Mouse Audubon's Coastal Waterbird Program, which works with coastal communities to protect rare birds and their habitats. We also have some workshops lined up. So if you're planning to attend our meeting, you have to make sure that you can if you want to attend also one of the workshops, that your flight is either early or late, because our workshops are planned for Sunday and Monday before the meeting starts, or Friday after the meeting starts. So we have one workshop on introduction to passerine banding, organized by the North American Banding Council and Manomet, and another workshop on Friday on educational outreach for field ornithologists, how to effectively engage students of all ages during bird banding and other field work. And you will soon be able to read this information about the workshops on our website and also register for them there. On July 29th, Our next AFO Café will be given by Maria Andreina Pacheco from Temple University, and she will be talking about avian hemoparasites, insights into avian evolution. Our AFO cafes are sponsored by Avianet Research Supplies, and I'm not sure if Patrick is here. I haven't seen him. I don't think he's here.
I don't think so.
No. Okay. So Patrick, Avianet is our research supply stores, and we actually provide mist nets and research equipment to field biologists. So if you need to purchase binoculars, mist nets, pistolas, or whatever you need for a field, please make sure that you visit our website, www.avianet.com. And if you enjoy our AFO Café and you're not a member, you can become a member today and support the continuation of these events too. And now I'm going to let Rebecca introduce today's AFO Café.
Hey, thanks, Valerie. So I'm really excited to introduce our speaker for today. I've been an avid Twitter follower of Jess's for several years, and I think that that's how a lot of us come together in this community. So I'm really excited to introduce Jess. Sorry, we have planes flying over. Jess did both their bachelor's and master's at the University of Alaska Fairbanks and is currently wrapping up PhD work at the University of Oklahoma. Jess is currently a postdoc at UC Berkeley working on the genomics of Caribbean anole ecomorphs, looking at the genomics of underlying that adaptive radiation. I'm sure Jess will come back to birds at some point shortly hereafter. But Jess's overall research focuses on using comparative genomics to understand broader patterns in the drivers of speciation, and especially looking to do this in the tropics. And so, Jess, we are eagerly ready to hear your talk, and take it away.
All right, thank you so much. And thanks again for the invitation to talk a bit about my work today. Let me just get this up. So I'm going to be talking about part of my dissertation work today, which most of my dissertation focused on looking at speciation in birds in Panama. And so I'm going to take you on a little bit of a journey with this work. And so at the core of a lot of my research interest is this question of how bird species actually form. And historically, a lot of our models of avian speciation have really been centered around the role of the landscape and of the geography of speciation, and really emphasizing that species arise in allopatry when they are isolated geographically and cannot hybridize with each other and start to develop their own separate evolutionary paths. This is kind of unsurprising that we're really big into this in ornithology in particular, since this was this really goes back to Ernst Mayr and his work. But the more we've looked at specific cases of avian speciation, especially as we've gotten access to more and more genomic data, that's allowed us to really look at the histories of gene flow in birds, we've kind of hit a problem. Turns out that speciation with gene flow is everywhere in birds. This is a screenshot that I did a while ago, and I'm sure if I went back now and updated it, it would be even more. But in fall of 2021, when I took this screenshot, just typing that into Google Scholar, found over 45,000 papers results for that. And so, clearly something that we're not capturing everything by thinking about things in terms of just allopatry alone. And this is further backed up by the fact that birds hybridize all the time. And as particularly people who work out in the field, it's entirely possible that y'all have actually seen some hybrids yourselves. Some of these came from folks on Twitter when I asked for pictures of different hybrids that they've seen. So clearly, we know this is happening, but if our theoretical framework of how species form doesn't take that into account, maybe we're doing something wrong. And there's a lot of ways to look at this, but kind of for simplicity's sake, because I am one chronically exhausted grad student and can't answer all of the questions that are wrapped up in that single question, I have chosen to really focus a lot on this question of reproductive isolation and why do some species develop reproductive isolation, but others never quite do. And there's a lot of reasons for this. One, postzygotic barriers and prezygotic barriers, like, things like, are these species chromosomally compatible and have the same number of chromosomes, versus do these species respond to each other, to each other's behavioral signals for mating? Those tend to actually differ a lot in effectiveness. So that could be part of it. We could have uneven divergence across the genome in ways that create incompatibilities, and those might be really hard to spot if you don't know what you're looking for. We could have varying strength of selection in different clades of birds, and these are all probably going on to some level or another in most systems. But why do they happen in some species but not others? And to get down to that, a lot of what I'm interested in is then actually turning that into factors intrinsic to the birds themselves, because birds are not just these passive things on the landscape that evolution happens to. They have ecological and life history traits that make it more likely that certain outcomes are going to happen versus others. So what can we do to actually explore this? And if the story is more complicated, how do we even start asking the right questions, let alone answering them? So a lot of my work has focused on secondary contact as a way to test for outcomes of reproductive isolation. Starting back in my master's, looking at Beringian birds, but increasingly I've looked at Panama as the system that to do this in. And if we think about it, if we have two allopatric populations of birds in eastern and western Panama, which is something we see quite a lot of, and I'll get into some actual numbers of that here in just a little bit. When they then later have range expansions, you can imagine that a couple different outcomes might occur. They could, like we see at the top, overlap in geographic ranges, but not actually ever hybridize and have complete reproductive isolation. They might, as we see at the bottom here, just completely collapse into a single population again that just shares all its genes and is panmictic across the whole space. Or we could have some sort of hybrid formed, anywhere in between those two outcomes. So what predicts where a given species is going to fall when this happens? So let's talk a little bit about Panama. And Panama, I'm sure a lot of you all know, is an absolutely fabulous place to study birds. And it's got 1,000 recorded species. And one of the things that makes it really interesting for the type of work that I'm doing is that since this is a very narrow landmass that between South America and Central America, it means that it's really easy to do a transect across a lot of different bird species ranges. And but as we were going into setting up this my dissertation work, And I knew that I wanted to really dig in on a few species in particular and do a lot of sequencing. You know, there was kind of an overall thing of like, well, we know from, you know, talking with, you know, folks like my PhD advisor, Matt Miller, and with our collaborators who've been working in Panama, that it's pretty clear that it was pretty clear to a lot of these folks that knew the birds of the area that there was a lot of diversity that we didn't actually quite recognize even in this 1,000 recorded species of birds. So if we're going to ask what's driving speciation, we first kind of have to know how many potential species there are. And I decided, well, if we're going to do that, we should also look at how some of these ecological factors play into that as well. And I apologize for the running commentary from the dogs. So let's get into some of the questions. So the very first part of my dissertation was just how common are cryptic species in Panama? And are some geographic areas more likely to have cryptic species than others? Just kind of getting a very general feel for what the avifauna of Panama actually looks like. And if then bringing in this question of, are there ecological traits that are more common in some of these taxa, that first looking at mitochondrial splits, and then bringing in more genomic data sets where we can get better measures of reproductive isolation and hybridization. Again, are there ecological traits or life history traits that are associated with greater isolation? So first thing I did was making use of this amazing barcoding data set that had been developed. I was saying before this went live, this data set was honestly started like, I think, well, I was still in like middle or high school. So long before I ever even knew that I was going to be studying birds one day. And had 2,300 individual birds from like 480 species included in this data set from all over Panama, and focusing primarily on land birds and those that were residents. So trying to exclude migratory birds and trying to exclude things like water birds, where you know, they definitely have stuff going on, but it's kind of the landscape histories and stuff that they're going to be defining, a lot of that are probably going to be different than things that live in forests, for instance. To maximize this, though, I took that bold data set, and then using some whole mitochondrial genomes that I'd sequenced as part of my PhD work, I was able then to call whole mitochondrial genome genotypes for Eastern and Western Panama for like 26 of these species. And with those, I was then able to go in and supplement further with data publicly available on GenBank. So this meant that while most of my data set was composed of CO1, for species, for some species, I was able to then also include ND2 and Site B and ATPA 6 and 8 for some of these ones where maybe we didn't have the geographical coverage we were hoping for with the initial CO1 data set. And with this data set, I was then able to start asking some questions. And the very first thing, this very first question, turns out cryptic diversity, it is, as we expected, really common in Panamanian birds. Of the 131 of the species were widely sampled across 2 or more geographic areas. And of those, 34 had, using this numerical taxonomy method of barcode identification numbers, they had 2 or more of these. And this is a lot higher than some of the other studies that have been out there. For instance, in North America and temperate zones, numbers closer to like 10%. So this is pretty high. As to where the splits are, there's a couple patterns that we noticed immediately. We'd expected going in that, you know, we're going to see a lot of splits in highland birds, because in Panama you have, you know, very, you know, a couple very discrete mountain ranges. And we did find a couple of these splits were indeed in the highland birds, but a lot less than we expected. And this is probably because a lot of those splits have already actually been recognized. Instead, what we found was most of our splits were in the lowlands. And so this is part of why going forward, I actually mainly am focusing on lowland birds for the second half of this talk. And we found a couple of different main contact zones that were showing up again and again in Darien, in Caribbean Veraguas and Cline provinces, along with a couple other patterns. The southwestern Burica Peninsula has a lot of unique haplotypes that are found there. And so, although we didn't, I don't get into that directly as much with some of this work. So there's a lot of splits. There's a lot more in the lowlands than you would expect, given the fact that historically, a lot of these are forest birds, that the forests were fairly well continuously distributed up until very recent times. So why are there all these splits in the forests and the lowlands? You know, a lot of these other ones make sense. There's geographic barriers, like, you know, for the highland birds or for the birds in the Southwest, where, you know, it's a population isolated it's a lowland population isolated by the mountains. So why are we seeing all these splits within the lowlands? And so this is where I started actually breaking down like things like habitat and diet. And one of the things right away jumps out. Understory birds and forest birds, way more likely to have these splits. Birds that are strongly territorial, way more likely to have splits. Birds that rely on animal food sources, particularly invertebrates, way more likely. And these are all statistically significant differences. And then finally, this is the piece that kind of ties all of those together. When we look at the hand wing index of these species, we find that for species that have splits, they have a significantly lower hand wing index than those that are the same haplotype across the whole region. And hand wing index, as a, you know, it's this ratio of that describes the shape of a bird's wing, and that ties in very closely with how well can these birds actually disperse, how well can they fly. And these other factors that we see are associated, they are also linked to lower dispersal ability. So with our first pass with this mitochondrial data, we can see that we have higher probability of these mitochondrial splits for understory forest birds, for highly territorial birds, for insectivores, for low hand wing index. These are all things that are associated with dispersal ability, with lower dispersal ability, which is a really interesting first finding. But again, this is just looking at mitochondrial data, and it doesn't tell us, while it gives us insight into some of these patterns, it doesn't allow us to really dig in on the extent of reproductive isolation in the way we would like. You're just looking at a single part of the genome, and we want to know, well, how much gene flow has there been? And for that, we switched over into genomics sequencing. So for this part of the study, I focused on 10 of these species that are widely distributed across lowland Panama. And I sequenced we sequenced ultra-conserved elements all across Panama, trying to build as complete a transect as possible. And we know that all of these diverged at different points of time. Because one of the other things is that when you're looking at things that are diverging in allopatry, there's this baseline assumption built in that if the longer it takes time for reproductive isolation to develop, therefore, the longer things have been separated, it's usually expected the more reproductively isolated they're going to be, the less likely they are to hybridize. So, and we had a pretty good idea of when these different populations had diverged between eastern and western Panama based on their mitochondrial distance. It's a pretty well established proxy in birds. And with this data set, we also decided to include 2 of these lineages where they are actually now recognized as separate species across Panama, but kind of as a control for, okay, we think these things, we currently classify all these things as conspecifics across Panama. We classify these as separate species. Are they really that different? So that's also another thing to note. And we can make some predictions from this. So if our kind of null assumption going in is that if time is the most important thing going on here, then as the pairwise mitochondrial distance increases, the width of the hybrid zone, which we're measuring with these Clines for all the for each locus, if the median Cline width is going to decrease as this pairwise distance goes up. We expect the same for the variation in the Cline width and center, where so basically, if you are looking at all these different Clines for different loci across the genome, the more we expect that over time they are going to coincide more geographically. The where you're going to find a hybrid based on one part of the genome is going to be the same as where you'd find it based on your expectations for another part of the genome. Again, this is going to increase, this variation is going to decrease over time. And finally, we expect that the proportion of the genome that is fixed, that is completely distinct between Eastern and Western populations, we expect that that's going to increase over time. And we'd expect this both on the autosomes and for the sex-linked loci on the Z chromosomes. Oh, did I just go backwards? Yes, I did. So, question is, do our data support these hypotheses? Nope. So we find that the average width of the hybrid zones, looking at median Cline width as a way to measure hybrid zone distance, it varies a lot. For our friends, the white whiskered puffbirds up here, they would literally fit like between here and my office on campus. 2 kilometers for most of this turnover to occur in. Meanwhile, with our orange-billed sparrows, we had to actually go back and sequence more birds from Nicaragua and Honduras to get the edges to actually get like pure parentals for the Western population, because the hybrid zone is so wide and gene flow is so widespread. So we have a huge range of hybrid zones, sizes. Well, but we had a huge range. We had a pretty huge range of divergence times. What's the problem here? Problem is they're just not correlated at all. R squared of 0, no relationship. But there were a couple other patterns that were emerging, and we'll get to those in a second. Again, with the fixed percentage of the genome that was fixed, again, no relationship. Variance, not really not a statistically supported relationship. So if it's not about the time in isolation, what's determining these outcomes? What is predicting them? This is where I went and ran a whole bunch of models and to try to figure out what other factors might be involved, if maybe time is important, but it's time in combination with something else. And then for the Clines, I went in and added some, I looked at the some measures of genetic differentiation, because if the genome as a whole is becoming more diverged between Eastern and Western populations, you'll expect, well, maybe there's more incompatibilities or more things under selection that are going to prevent hybridization. So that might be part of it. But not, even though that is a process that happens over time, it is not necessarily directly linked to time. So for the measurements of the actual geographic space we were talking about, I added in FST and nucleotide diversity is measured with pi. And for ecology, I looked at diet and hand wing index for all of these variables. And this, we did find some results. For the variation in center of the Cline, basically, how stable is the hybrid zone? Is the genome moving uniformly in introgression between these populations? Find that the predictor, the variation in that, is hand wing index, going back to dispersal ability. Meanwhile, for the how much the width changes, we found that FST alone was the best predictor. And as a note, I tested all of these like in combination with each other. So finding like single variables to be the best predictor was actually really surprising to me. So for the width of the hybrid zone, how much variation there is in the extent of different parts of the genomes being able to move, being able to introgress into the other populations, that's predicted by how well overall the genomes are differentiated, which makes a lot of sense because of some of the factors I already talked about. For fixation, these were both for the autosomes and the Z chromosomes. This was predicted by time in part, but also by hand wing in combination with hand wing index. So this makes sense. Time you need time for these variants to actually arise, but for fixation to actually be reached, it's not time alone. Like it appears that hand wing index, this measure of dispersal ability, is just as important a factor in parts of the genome actually becoming fixed. But for the width of the hybrid zones, this is where things get really interesting to me, because this is like the most, like, in a lot of ways, the clearest measure of, well, how much do they hybridize? What we find is that it's most clearly predicted by the diet type, whether they're insectivores or whether they're frugivores and granivores. And why on earth would this be what's driving the extensive hybridization? So there's a couple different possible things. So fruits and seeds, as those of you who work in the tropics know, like, they're common, but they're going to occur in clusters in space and time, as individual trees tend to put out fruit or seed kind of asynchronously. And this is backed up by the fact that in general, frugivorous and granivorous birds in the tropics have larger home ranges than those that are relying on arthropods. And arthropods, they have a much more continuous distribution in the forest in both space and time. So we know from previous studies, insectivores, they tend to have these smaller home ranges and lower dispersal ability. So it just comes back to dispersal ability, right? Like, clearly that's what this is all about. One problem with that. Remember, I included hand wing index as a variable for these models. And this is a much more direct measure of dispersal capability, but it's not included as a variable in predicting Cline width. So this brings me to my hypothesis that I'm currently exploring when I'm not wrangling lizard DNA. Currently, I have sequenced some more genomes for these that just didn't get included in my dissertation, because I think that one of the other big thing that might be going on here is diet and its link with overall demographic stability over time. Frugivores and granivores are more likely to have fluctuations in range and population size driven by these boom bust cycles of the plants they rely on. And while insectivores, they are also susceptible to this to an extent. It's not experimental evidence has found that it's not to the same extent as to those birds that rely on these plant-based resources. This does require more data for fine scale modeling of population size change. The one problem with the ultra conserved elements approach that I used here is that ultra conserved elements are these markers that are anchored by, shockingly, a highly conserved portion of DNA. So while they're really great for doing these sorts of studies in species that aren't particularly closely related, because you're comparing the exact same parts of the genome, it means that if you want to get a really fine scale model of time, they're not very good for that, because again, they're conserved. They're not going to show the same signals of demographic change at that model that we need. So this is this is where I think probably a lot of is actually driving some of this. So in conclusion, there's lots of unrecognized diversity in neotropical birds. And while some of this is potentially driven by observer bias, like we saw with, highland birds probably being underrepresented because they're already recognized as separate species, it does look like ecological trends do support that play into dispersal capability, and other factors play a key role in actually generating diversity. In ecology, it's not only tied to the likelihood of diversification, but to the maintenance of reproductive isolation when things come back into contact. Time and allopatry, it's not the primary predictor of whether or not things hybridize when they come back into contact. These other factors, particularly diet, seem to be much stronger predictors of that. And so I hope going forward, this kind of provides some bit of a different framework for thinking about how we think about speciation, and not just kind of defaulting to just reaching for geographic explanations of why we see the patterns of diversity that we do, and that these ecological factors are just as important. So just for some quick acknowledgements, most of the specimens for the study came from STRI in Panama, as well as from the University of Alaska Museum and from the Berkeley Museum. And thanks to Mi Ambiente in Panama for collecting permits for most of these were museum specimens, but I did do some collecting back before the plague in Veraguas to cover what turned out to be a really important gap in my transects that ended up being like right in the middle of the contact zones for some of these. Also thanks to the Sutton Scholarship and the Alumni Fellowship at the University of Oklahoma, which provided support throughout my PhD to do this. And then to my collaborators, Matt Miller, who was my PhD advisor at OU for a good chunk of my degree, but then to the many, many people who contributed in some ways, particularly to the barcode part of my of this. Like I said, this is a data set that was started long before I was in science. And so while I kind of did a lot of the final work of tying it together and coming up with some hypotheses to test with the data, the actual generation and making it happen of the data is goes to the many other people. And also shout out to Jorge Garzon, who was helping me in the field when I did go to Panama, and helped dig me out of what I guess was probably quicksand. I just tell people it's quicksand because that's cool. So with that, I will take any questions that you have.
Thank you very much. That was really interesting. Thank you. So now I'm going to ask everybody to turn on their cameras. I mean, if you want to, and if you have questions, you can raise your hand or type on the chat box and we'll read those questions. And I will let people think about the questions, and I have a short question for you. So you looked at insectivores, but I'm assuming that these are gleaning, like forest gleaners, or not aerial insectivores, right?
Yeah, correct. For the most part, in I don't know if I can get back to the slide that had it. I did have a couple of aerial insectivores included in the barcoding portion, but as you can expect, they kind of follow completely different. They're
Yeah.
kind of doing their own things, but yeah, all of the ones for the UCEs were completely and I think I even coded them differently in the data analysis, although I'd have to go back and double check. I only had like 2 aerial
Yeah.
insectivores.
I'm just wondering, I don't know, I mean, if there are any examples of that, but if you could look at aerial insectivores that are dependent on, like, you know, that are close to wetlands and are dependent on those pulses of insects the same way that frugivores would be dependent on the pulses of fruits, you know, or something like that. And so maybe that might give you an idea of it's the pulse itself
That's a good idea. Yeah, I hadn't thought of that. Mainly because, like, shockingly, a lot of for a lot of these specimens, the forest understory birds tend to be a lot easier to get samples for, since they're a lot easier to catch. But that's actually that's a really good idea, because that would definitely be a similar pattern, and show that's like it's not anything special about insects per se. It's the demographic and other factors tied to them. So that's a good idea.
Well, Kevin Bennett, you have your hand raised.
Thanks. Thanks for a great talk, Jess. You mentioned that you included some splits that were recognized as splits in addition to the conspecifics. Did you have enough power to kind of see if the patterns were sort of the same for those comparisons versus the conspecifics?
Yeah, so I didn't do like a direct comparison, because again, only 10 species and 2 of them are recognized as splits. One of them's the Chiffornis, which has only relatively recently been split. But particularly, let me pull this up. In terms of just the overall impression, and, you know, again, don't have statistical power to say for sure, but like they're pretty middle of the road, honestly. Like, they're not there were a couple of results where, like, the fixation rate for Chiffornis on the Z chromosome was like way higher than anything else, but like for the rest of it, they look pretty identical to these things we consider conspecifics.
Thank you. Dan Baldassari.
Yeah, really cool stuff, Jess. And I was wrangling a 1-year-old during a lot of your talk, so I might have missed some of this, but and this related to Kevin's question. You're talking about cryptic diversity, but is there, in any of the species that you did these transects on, is there any phenotypic sort of variation that co-varied with any of your genomic stuff, or are they really, if you just looked at the skins, really, totally, monomorphic across that range? Or is there any hint of, some phenotypic divergence?
There's some, but it was hard to figure out how to quantify some of it. Bayrens, like, it's really clear that yes, there is a difference. That's like the main one that really stands out. There's some difference in the Henocarina wood wrens. There's some difference with the puffbirds, but it's kind of at the level where and unfortunately, this is a section that I wasn't able to pursue due to, the pandemic hitting. I was hoping to spend some time at Stry actually figuring out quantifying some of these plumage differences to get into that, because while for some of them is like, yeah, it looks like there's a difference. It was the sort of thing where it's like, is there actually a difference, or am I seeing one because I now know that there's a difference? And so, you know, I think for a lot of these, there's probably some level of difference there. It's just not enough that, because humans tend to not, we're not birds. We're not looking for the same things they are. So, it could be that there's something that we're not noticing, particularly getting into the stuff with the barcode stuff. Like, there's probably some, for some of those, there's probably some probably obvious signs that just got overlooked or chalked up to, like, quote unquote subspecies level differences. But it's all stuff at that level. Like, the only one that has the Ramphichelys tanagers in that chapter, they're really easily distinct, but also they're like the ones that are included because they're two different species, and I want to include them. So, like, everything else is like, with the exception of the bay wren, it's like maybe, but again, didn't get a chance to actually quantify that because all the specimens were not where I could access when we were all quarantined.
Yeah, it would probably be a whole other dissertation just to quantify all species phenotypically, right? But it does like something that would be a really interesting, like, extra, layer on top of the genomic stuff, right? To look at how well those things correlate and if there's anything there phenotypically.
Yeah, and I did start with because there's additional data sets that I'm hoping to, after I'm done with my little lizard vacation here. I sequenced whole genomes from eastern and western Panama of 26 species. So these 10, and then a bunch of others. And I did start on preliminary, like, morphological measurements. The last time I was up in Alaska, where some of the specimens are held. But and the plan was to incorporate some of those and to try to bring in some of the stuff in terms of, like, colors and the spectro stuff and whatever. But a lot of it just again ended up not being able to come together. But the long-term plan is to include some of that in the analysis with the whole genomes down the road. So it's just yeah.
Thank you. Ghislaine, you have a question?
Yes, well, I really liked your presentation. I think you're really, really onto something here, because I've done at least 5 years of research in central Panama, and there's always something that comes out as to the possibility, if you look at the different forest fragments nowadays, how some birds just cannot cross, like the BCI, for example. Just those species are like very, very specialized to the understory. They could be blinded by full light, full sun. The very, very short rounded wings don't allow them to fly stuff for a very, very long distance. So I think you're really onto something in looking at, and we never thought of the speciation and morphology, because it's really going to bring something interesting, and it might actually link to the conservation of all of those understory birds that we have today in Panama. So I think that's pretty good. I had a quick question. Do we have a history with all the people that try? Do we have a history of how forests moved around, or if we think it was forested since when the canal, no, the Panama, the isthmus was formed? I know there must have been some movements there, but is there records of that, or?
Yeah, there are some records, particularly pollen records. And there's definitely cycles of forest expansion and contraction, particularly with like grassland spreading through more arid periods of time, at least for the Pleistocene. I don't know if we have really good records going all the way back to the formation, just because, you know, that's a long time to get a pollen record for. But while there seem to have been definitely periods of aridity, and it looks like at least there's some localities where it looks like forest was probably pretty continuous throughout that time. Some localities it looks like maybe there was more of a cycle, but and it's actually pretty hotly debated as to just how continuous stuff has been. It looks like there's probably always been, you know, going on some of the pollen stuff, it looks like there's probably always been at least some forest, but you know, just it you know, some of that's still a bit of a question mark.
One more question quickly. Do we have records of bird measurements? How far back are we going? Did Chapman did anything when he was on BCI in the 30s? Was there any data that could be, is there any changes that could be seen from the earliest data we can get, or?
In terms of like morphology and stuff,
Yeah.
I'll admit I haven't looked for that in a lot of cases, but I believe there's been at least some, because there's, you know, a lot of the stuff has come from studies where people have went and looked at some of that. But as to exactly how far back they go and if there's like a good time series, I'm not really sure on, but you know, certainly the specimens could some of them could be measured for some of these things, but you know, depends.
Thank you. Kevin, you have a question. Yeah.
Yeah, I just wanted to just following on the issue of the sort of historical reconstruction of habitat. This is not my area of expertise at all, but I was recently at STRI and talked to Aaron O'Day there, who has done some really cool like coastline reconstruction stuff, and I suspect his lab probably has some good resources on that.
Yeah, I've definitely cited quite a few of theirs, their work in my their papers in my dissertation. But yeah, no, that'd probably be good for future stuff to look more into what they're doing.
Yana, yes.
Yeah, hello.
Hello.
Yeah, thanks for the talk. Very nice. So I was just wondering if, maybe you mentioned something about this, but did you look at any differences in males and females, maybe sexual dimorphism in color or song, maybe only the males sing, not the females, just to get at, for example, sexual selection, pressures and such, and to see how it matches with your Cline variation?
Yeah, I was not able to do that just because for a lot of my sample sites, the vast majority of my samples were museum samples, and you know, obviously they tried to record like male versus female on a lot of them, but in you know, it was harder to go and like yeah, I didn't get a chance to do some of the stuff in terms of like playback and selection directly in the field. Again, it got COVIDed with a lot of the rest of the field component of my project, but and I did do some exploratory stuff as to whether or not we could look at any differences by male and female, but unfortunately, a lot of we did have a pretty strong male bias in the samples that we collected, so we just didn't have the statistical power for a lot of our sites. So, you know, if we only had, for a lot of our sites, we only had like 3 or 4 samples, and all or most of them were male. So I just didn't have the power that I'd like to look and see if there was sex bias in some of this. So, but it's definitely
Yeah.
something I'd love to get into.
Well, sorry, so I think I wasn't very clear about the questions, but I just meant maybe in looking at plates in the Birds of Panama book, to
yeah.
look at the differences and then related to, I guess, this pattern of divergence that you see in some but not others.
Yeah, for these 10, these are all pretty monomorphic. As to the larger data set of with the mitochondrial stuff, I should go back and check that, because I didn't think to do that.
And also because I think some studies have shown that it might relate to dispersal abilities. You know, for example, if in polygynous species, you know, sometimes females travel pretty far to find them, to search for mates. Yeah.
Yeah, no, and I
It
think
would be interesting.
yeah, and I think some of that might play into like you know, the pattern that I saw with the territoriality. I would need to dig, yeah, I would need to dig back more into that and look, because you know, it might be interesting to tease out like if, you know, what exactly types of territoriality are playing out, like if you have, you know, both sexes defending territories, or if you've got a lecking system, or if you've got like strong male defense of territories, and see if that makes a pattern. That's a good idea.
That sounds good. Yeah, thanks.
Yeah. Okay.
Thank you. Stephen Rothstein, you have a question?
Yes, thanks for a really interesting seminar that reflects an enormous amount of work. Early on in your talk, you mentioned that the allopatric speciation model is the prevailing model, at least has been for quite a while. And then I got the impression that you were going to show that that's not the case. And it seemed from your results that you clearly show that time alone is not going to determine genetic divergence. Other factors are involved, but I didn't see something in your results that would show that anything you found is not started by some allopatry. Am I interpreting things correctly?
Yeah.
At least to your viewpoint.
Yeah, and I think I think the key thing and how the allopatry thing plays in here is that at least a lot of the formulations and like how you would test allopatry are very much founded on the assumption of time being a driver. And so I think in the Panama stuff in particular, I don't think we can necessarily, I don't think we can disprove allopatry as a thing. And in fact, I think we have some strong support that allopatry is driving at least some of the initial differentiation, but it's not what's necessarily driving reproductive isolation. So, and I think the key thing is, for me, is disentangling those parts. And you know, in that if we just assume that it's just this very strict look at allopatry instead of, well, allopatry plays a role, but we also need to bring in these other things, that's kind of the key difference to me. Sounds like I probably need to make that more clear though when I'm talking about it.
Yeah, thanks. That clarifies things.
Yeah.
Thank you. Do we have any questions on YouTube, Matt, or I don't know?
No, not on YouTube.
No questions. Are there any other questions here? No? Well, thank you. Thank you very much, Jess. That was a very interesting talk. Thanks a lot.
Yeah,
And,
thank you for the invitation.
Good luck with the future research.
Thank you.